So have you heard of this new study where they were able to take human skin cells and turn them into eggs to help women with infertility? This is what's called in vitro gametogenesis, or creating gametes, which are the human sperm or egg, in vitro or in a petri dish outside of the human body. This can be really valuable because one of the reasons that women have issues with fertility is that their ovaries cannot produce viable eggs, and that might be due to chromosomal problems, problems with the ovaries, women who've had chemotherapy and damage to the ovaries, or most commonly women who are older, and with age the ovaries don't produce as many eggs and as many viable eggs. So if you could produce an egg outside of the body, then the rest of the steps that are taken in vitro fertilization can be used to then fertilize that egg and create a viable embryo and a viable baby down the road. Initial attempts at doing this over a decade ago focused on stem cells, where they took stem cells, which are these cells that are not differentiated or haven't decided what kind of cell they're going to become and have the potential to become any cell, and exposed those stem cells to certain chemicals which made them differentiate into an egg. And they showed that in mice they could effectively do this, and those eggs could be fertilized and lead to viable mice. The problem is that in the decade of research, they haven't been able to do this with human stem cells, so that approach to creating eggs so far has not been successful. This group took an entirely different approach. Their approach is actually somewhat similar to the approach that was used for cloning years ago, when in Scotland they created the first clone of a large mammal, which was Dolly the sheep. And in that case, they took an existing egg, so they had the structure and environment and chemical contents of that egg already there. They took out the nucleus, which contains the genetic material, and they replaced the nucleus in that egg with a donor nucleus, in this case from Dolly the sheep, and then they implanted that egg with Dolly's nucleus, and it was able to grow into a full fetus, which became the clone of Dolly. So in this case, they used a similar approach, and they showed that a few years ago that this actually works in mice, where they take an existing egg, remove the nucleus, and then take the genetic material, in this case from a skin cell of a donor, because remember, all of our cells contain all of our chromosomes, so all of our genetic material is there in every cell, and they took that nucleus from the skin cell and put it into that egg. Now, the big challenge was that a gamete, or an egg, should only have half the chromosomes of a normal cell, like a skin cell, because remember, the other half have to come from the sperm. So the big challenge was to get that egg to then shed half of its chromosomes so it could be like a normal human egg. They showed that they were able to do that with mice. The egg shed half of its chromosomes, it was successfully fertilized, and it became a viable mouse. This study, published just Tuesday, is their attempt to do this in human cells. And what they showed was that although the same technique that they used in mice did not work in humans, surprise, surprise, they were able to find a different technique to effectively get many of those eggs to shed half of their chromosomes, and in some cases lead to possibly viable fertilized eggs. Now, in most cases, this did not work well. When the eggs shed their chromosomes, they did it in a haphazard fashion. It often wasn't shedding the right number of chromosomes, so they ended up with the wrong number of chromosomes. Even when it was the right number of chromosomes, the chromosomes did not recombine in the same way as we have recombination of our genetic material when we form gametes and normally in the human body. So those gametes were not reflective of the full genetic material in the donor. So there are many steps that remain to be sorted out here in this process. So there's a lot of buzz about this because it's an interesting and completely different way to address this issue of creating gametes outside the body or creating eggs outside of the body. But it's also very early. There are many kinks still to be worked out, and we are years and years away from this potentially being an effective way to address female infertility. Also, some ethical quandaries here. They're using the same technology that was used for cloning. They're doing research, if this is successful, ultimately research on viable embryos, which is not allowed in many parts of the world. There are concerns about eugenics. There are concerns about being able to control the genetic material that goes into these gametes and being able to then manipulate what the genetics of the offspring look like. But overall, a complex and amazing body of work, and a reminder of how incredible science can be.